Is this on my side? Why? I'm hitting, oh, there we go. I wonder if it's going to advance several times now. No? Okay. So this is, I know many people here are going to be more familiar than I am with this, just to make sure that we're all on the same page. Long QT syndrome is an arrhythmia that predisposes people for torsade de pointe. And so here is a surface ECG, which voltages are measured on the chest of an individual. And these features reflect the depolarization and the repolarization of the tissue that composes the heart chambers, the various heart chambers. And if we look at, say, a representative cell within the ventricular chamber here, it has a resting state at -40 millivolts. It receives a signal to depolarize, which it dutifully does, and has this characteristic shape, this plateau, and then a precisely defined interval at which it then returns back to its polarized state. And within that interval, the contraction happens. And so now the profile of this action potential and the duration are controlled largely by 3 ion currents, 2 potassium and 1 sodium. There's a calcium current in there as well. But these are the major genes that are associated with the genetic phenotype or with long QT syndrome. And as you might imagine, the interval needs to be controlled precisely such that if there is a prolongation in this interval, the cell may be refractory to receiving another signal again, and that will result in problems in terms of the conduction across the ventricle. So, all right, so we know that there is a genetic substrate for this individual, Mrs. X, for her disorder. It looks like there are 2 candidates, at least when this was being evaluated. But what that really points to, as far as so she, she had beta blockers, she had an ICD, she was being monitored. But what it really indicated was that, there might be more individuals within this young woman's family that are going to be at risk, and so they need to be evaluated. And so here's the family tree, and these are the individuals that are, looked at, which I'll show in a moment. I'm going to rush through this. It's been published, and I've gone through it a few times. But here are the 2 variations that came back from GeneDx commercial testing lab. Both of them were called pathogenic, and they used these data to go on. And so it's, KCNH2, variants within KCNH2 are responsible for 30% of genotype positive LQTS cases. It was found, this particular variant was found in 5 other patients that also had LQTS. In the area around this variant, there are variations that are also associated with long QT syndrome, and it's rare in the population. We have a similar set of evidence to support the pathogenic, at least at that time, support pathogenic classification of this other variant that this young woman carried. Variants in KCNQ1 are responsible for 50% of genotype positive cases. It was found in only 1 person who also had long QT syndrome. There are variants in the vicinity of this variation, if you look in sequence space, that are also associated with long QT syndrome, and it is rare in the population. And so we did, some functional characterizations. I'm not going to go into the detail here, but the bottom C and D are a functional characterization of this KCNQ1 variant. The open circles are wild type. The black circles are the variant, and you can see this particular functional measurement. They overlap quite nicely. Same thing on top, only this is the hERG variant, and you can see they diverge tremendously suggestive that the hERG variant is much more disruptive, at least in terms of this functional assay, than the KCNQ1 variant. And here's looking at sort of the structure to try and evaluate a little bit closer the context. So this third point that is used as justification for the classification, you can see is a much more densely packed area here in hERG, KCNH2. There's a more densely packed area, and the residues that are also associated with long QT syndrome are much more close to the residue of interest here. And you find it a little bit more spread out and more heterogeneous in the case for KCNQ1. And so these are, so these are data, they are relevant data. But as I'll point to in a couple of slides, I think the real meat on the story is, well, there's a family, and so you can actually just go in and find the genotype of these people, and it happened that there were several of them that had either one variant or both variants, and then you can look at the clinical phenotype, which you don't always have that opportunity, and so looking here, QTC, a corrected QT interval without any secondary cause for prolongation over 480 is in some circles substantive enough for the diagnosis. And so you can see here those individuals in the family, this is the proband again, that have the KCNH2 variant or both have QTCs that are on the edge or above that particular threshold, arbitrary as it be, whereas the KCNQ1 carrier, the sister of the proband, and her two kids have both are KCNQ1 positive, and have a much more mild phenotype, if anything. And so, this is, so the issue here is that these results were, well, okay, so one, as the story goes, the family was given both the pathogenic assignment of those variants as well as their genotype information. And as you might imagine, the proband having some pretty striking presentations, the family was very concerned about her sister. And so really wanted to have an ICD and more active, management strategies for the sister. Vanderbilt being more savvy, folks in the arrhythmia side suggested that the weight of the evidence was not good enough, strong enough to classify both of them, but rather probably just the one. And which would be the KCNH2 variant, and the family, as you might imagine, was pretty upset that their desires were not being honored here, and so they left, and they found someone who was willing to perform the procedure, and one of the nurses here contacted that individual and asked, was there something we missed? It didn't seem like it really warranted that direction at the time, and the response was she had a pathogenic variant. And so that's it points to a pretty serious outcome that affects not just the initial proband, not just the sister either, but you can imagine this will flow generationally. They're aware that this variant is present in their children, and then they have a risk of passing it on to their children. So it's a very rich and personal field to try and dive into. And I kind of provocatively state this, is the family actually better off knowing their genotype in this case? And that it does feel like a decent amount of the strategy today is to move towards ignorance. So this is a case report. This is a specific example, but it's representative of a large issue. which is, as more individuals are sequenced, the more we sequence people, most of the variants that we find are rare, somewhat paradoxically. And here's an example from 50,000 exomes. And along the x-axis is allele count. I want you to read, people. Along the y-axis is number of sites, and I want you to read, unique variants. And so this sort of matches intuition in that there are relatively few, and they're both on log scales. And so there are relatively few variants that you see in a lot of people. So there are relatively few common polymorphisms. But if you look at the majority of what comes back in terms of the variants found, the overwhelming majority is an N of 1. So this is on a log scale, so it's pretty blown up. And so, it's what I presented just a few slides ago about the family, having a few carriers is somewhat of a luxury. For most variants, we're not going to have a lot of clinical data, and I would argue we never will. So our ability to observe variants in individuals is obviously limited. So what strategies do we have? So the ACMG, American College of Medical Genetics and Genomics, has a guide to variant classification. And I will buzz through this because I think many people are already familiar. And, it involves the data that I sort of presented before. So you have variant-specific information concerning the functional properties, the silico predictions, and where it's located in structure or in sequence, and then what's the carrier data, or people that are heterozygous for these variants. And you can collect these data and come to an assignment, which I've sort of represented here for these 3 variants that are in KCNH2. I've argued, even with Dan, about the classification scheme having having the appearance of determinism that I think is unwarranted. I mean, you think of these genetic influences as probabilistic in nature, shifting maybe towards really high or really low. So in practice, benign versus pathogenic in many instances would be appropriate, but it is in my, not sufficiently appreciative in terms of language about the probabilistic nature here. And so, you can break the words into their etiology, benign meaning gentle and kindly, and pathos meaning suffering, disease producing, which really, conveys a determinism. And so you can imagine if you're going to stick things into deterministic buckets, you're going to have a pretty high bar for moving them one way or the other. And in a lot of cases, that's not reached. And so this is, looking at the number of variants. This is several years ago now, but the trends are going to be very similar, that the majority of variants pile up in the US category. So as I sort of hinted at before, if I could get any data of my choosing, I have a structural biology background. In Dan's lab, I learned a bit of the functional side of these ion channels. But, if I could choose what data were available to evaluate, and especially in the clinical context, it would be just other carriers. Just find the variant of interest in the literature or in various databases, and I'd want to have deeply phenotyped EHR histories, including these records of gain of function and loss of function arrhythmias, at least to evaluate variants in the ion channel genes that I mentioned at the onset. So now I'm going to go through. So then, all right, so we're faced with, all right, well, how close can we get to this, ideal? And we're going to start with these 3 ion channels. There's it's a strong Mendelian link between LQTS and these 3 genes. They're ion channels, and so the functionality of them have been, tested over many decades now. And there are actually a decent number of variants that you can find in the literature. I should say this was a really frustrating exercise for me because we know that there are a lot more. Like it's like they're all kind of siloed off in a bunch of, you know, distributed centers that have their, you know, their own little fiefdoms that hold these data, and you know, for good reasons. But it's frustrating from a basic science perspective, because I know there are more out there. And it was actually, you know, I reached out to a good friend of mine, Joe Ali, in Paris, because I was so upset trying to find search through the literature to find these variants when I know that they're more accessible. And he helped me kind of get in touch with this hospital, this Bichat hospital. They've been great collaborators with us. And so, you know, we ended up taking all variants that we could find from the literature, but then also being able to augment that list through our collaborations, you know, help through personal contacts, but also through Leduc funding that we've gotten, which I've been able to participate through Bjorn. And okay, so this is, you know, a bunch of these are published. This is actually in revision right now. These are the individuals that were, you know, sort of led the charge to do this effort, which you might imagine is pretty tedious. Okay, so I'm going to bounce around a little bit back and forth here, but I'm going to try and highlight, you know, or point out when I do. So this is looking at KCNH2 variants, and this is just to sort of calibrate or try and calibrate the data set to your intuition, which I think people would intuit this is what you would find, which is you find a decent number of variants, and you find that I mentioned on the previous slide, you find some that are mostly in unaffected individuals. So this is a you know, a polymorphism, a pretty common polymorphism. You do find that represented in some of the loss of function. Here, that's going to be long QT syndrome. So this is, you know, an ion channel gene, loss of function, prolonging the QT interval, and then gain of function, a hyperactive potassium channel actually being associated with short QT syndrome. And then you find some variants where there's very few unaffected individuals, and then there's, you know, there's a strong bias towards the loss of function clinical phenotype or the gain of function clinical phenotype. And so this is a lot of individuals to sort of draw from to try and understand what this variant might mean to an individual that you're seeing, maybe agnostic of any of their particular clinical history. The problem is that there are variants like this, which is you have a single variant and you see it in one person, and that person may or may not have a phenotype. And the challenge is that the majority of the data set comprises these types. I've learned recently I say it wrong, and I'm trying to correct myself, but it's challenging. Okay, so we're limited fundamentally by the number of individuals who carry these variants. So, you know, we will find a lot of them. There are a lot more out there, I'm sure. It'd be great to have access to everything, although there are, you know, practical challenges there. But we do have a hard cutoff. So what can we do to try and enhance this understanding that we observe from individuals? And so this is now looking at SCM5A. Now the molecular loss of function phenotype is a loss of sodium channel current, and that's associated with this arrhythmia Brugada syndrome. But I just want to illustrate the point here that if you look at variants that have been functionally characterized, and you look at this molecular phenotype, which is the overall current that's produced. So we're going from something that's pretty much wild type here, which is a common polymorphism, as you might expect, down to something that is complete loss of function, which is this variant here, which has a strong association with Brugada syndrome. And what we see is a trend that this percentage of individuals, or penetrants, which I'm going to call penetrants, has an association. There's you know, a directionality that you might expect, and if you calculate the penetrance, loss of function penetrance here, Brugada syndrome, and then you look at, you know, this functional characteristic or some in silico measures, and you do a rank order correlation, what you find is that there are actually several of these parameters, as you might expect. You would expect some of these in silico predictors to be able to correlate with the with penetrance or with the prevalence of the disease. And so, all right, I want to make sure that I'm not taking too much time here. Okay, so there's a significant value there. Now there's a lot going on here, but I just want to walk you through just a couple of pieces. So this is looking at the same data here for KCNH2 and KCNQ1, functional parameters towards the top and silico parameters towards the bottom. And this is looking at, this again, this Spearman rank order correlation. I'm just going to point out a couple things. This is looking at correlation with long QT syndrome. And on this forest plot, I want to point out there's a lot of associations that are sort of dubious that cross this 0 line, but then there are a couple. So homozygously collected IKR, heterozygously collected IKR, peak tail current. So this is IKR, and here's the steady state current, which is sort of this first part of the current profile. And then here's the peak tail current. And so what this is saying is that peak tail current, as that goes down, there is a higher association with long QT syndrome, which is the directionality we would expect. And then for KCNQ1, so that produces the IKS current here, which has this profile. There's peak current, which is this, the maximum current that it reaches, and we see the same thing. It's negatively correlated with the presentation of long QT syndrome. Okay, now we can also look at the density in the protein structure. So you can look at where are the hot spots in terms of the sequence, but you can also map that onto the structure. And what we've done here is take a position for each position. We look at what's the penetrance of long QT syndrome in a sphere. So it's really like a penetrance density, and we've mapped it onto the structure. And I mainly want to highlight that it's heterogeneous, although we do see much higher prevalence or higher density of long QT syndrome in regions that you might expect. So this is where the ions go through this pore here. These are the voltage sensors, which are much more heterogeneous. And then there's some of these intracellular domains that are considered hotspots. But the main point I want to convey here is that it is heterogeneous in terms of, some parts have very high penetrance density and some do not. So this is looking at the same sort of thing with KCNQ1, and I'm just going to sort of rush through this, but it has very similar type associations as you might expect. One of the differences here is comparing the selectivity filter and the pore for this is KCNQ1 and panel D, and E is KCNH2. And you see there's actually there's a lower penetrance down towards the intracellular face compared to KCNQ1, which is suggestive that some of the nuanced interactions here and the intracellular face of the protein are much more sensitive to substitution than, say, KCNH2, which is an interesting finding. So here is looking now at SCM5A, and this is the penetrance density or the density of Brugada syndrome up in the top, and the density of long QT3, which is the gain of function. And so why I wanted to put this here is just to remind folks that the loss of function is associated with the peak current going down, but this ion channel is unique in the sense that it opens, but there's a fairly finite window here for this cardiac sodium channel. And there's a sharp inactivation that happens. And so if the inactivation happens too slowly, or if it doesn't happen fully and you end up with a persistent current, then you end up with a gain of function phenotype clinically, which would be the long QT syndrome type 3. And I mainly wanted to point out that if you look at the density of residues that are more densely associated with high long QT type 3, you end up with this sort of clustering in this intracellular space here. And this makes sense because this is really where the inactivation takes place. And it's controlled in this region, and especially by this domain 3-4 linker, which you can kind of see along here if you're following my laser. And so that sort of matches up with intuition is in terms of what we would expect. And then it's sort of the inverse of that that's associated with the loss of function more heavily, the loss of function phenotype, and it's more heavily, it's more dense towards where the selectivity filter is, as you might imagine. So I'm going to not spend too much time here, but we've tried to develop a method where we use a Bayesian strategy to take these data, which are things that we can get, and try and impute something that we can't always get, which is what's the penetrance. And so we want to calibrate that based on what functional or what data are available, and what variants where we have high confidence in the penetrance that's measured. And it's estimated. It's always estimated, but we try and get closer and closer to a better estimate. And that's what we use to calibrate how we interpret these data, which are available, this in silico structural and functional data. And so we end up converging on a prior that's informed by these data. And this is, I'm going to rush through this again. Two's been published a little while ago. This is looking at some evaluation metrics for KCNH2 and KCNQ1. Those of you who know me know I really don't like ROC curves applied in this area because I don't feel like it's appropriate. ROC depends on capital T truth being binary, which I don't believe it is. However, we've adjusted the evaluation here to only look at n=1 variant. So a variant that is only seen in a single individual. And in that case, they do either meet the threshold for diagnosis or not, according to our scheme. And so then our ROC is appropriate here. And this Bayesian strategy, we've actually gone through several different names. Penetrance, as Dan will tell you, is inflammatory in many circles. And so we used here in this paper post-test probability estimate or Bayesian prior here in KCNQ1. And so you can see the performance here. And we split it out into literature and cohort for this particular paper, and you can see it does reasonably well, I would argue. Now, if you accept the premise that this is continuous and probabilistic, then the ROC, as I argued just a minute ago, is not the correct metric to be using, and a Breyer score would be more appropriate because it's an evaluation of a probabilistic assertion. And here is some intuition, just a single example, and this is where the context in which I became familiar with it was evaluating statistical models to predict weather. And so you have an issued forecast and gives a certain probability, say 70%. And then if it rains or it doesn't rain, you end up with a Breyer score that's much higher. If it doesn't rain, you end up with one that's lower if it does rain. And so as you get closer to zero, that means your model is more well calibrated in a probabilistic sense. And so you can see here the Breyer scores go down tremendously, and they don't change much when we do tenfold cross-validation. So we think we're driving. I don't really want to argue that this is like the best method ever, but I think we're driving in a reasonable direction, and I think it's these data are indicative that what we can observe and what we can do by looking at these variants and the clinical data we have at hand is consistent with a more probabilistic strategy. Might not end up being this one, but I think that this is a reasonable starting point. So this, I think, conforms with intuition as well, that if you plotted just the observed penetrance, here looking at LQT type 1, which is variants that are in KCNQ1, you end up with a lot of variants that are seen in maybe 1, 2, or 3 people, and they're all of them do not have the phenotype, and then you end up with a bunch of unique variants, and that's what's along the y-axis here. where you see it in only one person or two people and they're both affected, or three people and they're all affected. So you end up with this sort of piling up at these either extremes. And this is sort of the ascertainment bias sneaking in. But if we can leverage our understanding of these variant specific features in silico, functional, structural, we can temper those expectations based on how we think those attributes associate with the clinical phenotype. And so this is what that ends up looking like. So if you look at unique variants in KCNQ1, KCNH2, and SEM5A, and here, the penetrance estimates along the y-axis in this Bayesian formulation, and then we plotted a couple of, or Matt plotted a few variants and gave their specific details to sort of point to, when you see 45 affected out of 123, and that's actually probably a little high there. And again, this is another, there are more people out there, and I'm sure it should come down to, I think, 20% or so. But it's not 100, it's not 0, it's not a risk factor, it's a substantial elevation. There are other things at play, obviously, but I think it's a disservice to treat this as somehow worse or equivalent to something where you have a much higher penetrance expected. The last thing I'm going to point out, well, okay, two things. One is that, this has the distribution that you might expect. It's disease specific, and so you don't expect a lot of variants here in SCM5A. It's a bigger protein. And it's responsible for a far lower percentage of LQTS cases. And so you don't expect to have a huge number of these variants having very high penetrance. And then you see that similar, that distribution change as both the size. KCNH2 is is actually a lot smaller. It's closer to KCNQ1. Q1 is the smallest, 650 or so residues, whereas this is 1,100 residues, but also responsible for a different number of cases. The last thing I want to point out is that this peak right here, if you're following my cursor, should be close to 0. This is, I believe, wrong. And what this is indicating is that our predictive features are not good enough. The predictive features we have cause too much of a spread here. And so this is sort of the average that comes out based on that variance with a C in those parameters. And so here, I think there's actually a decent upside if we could get functional data. Hint, Dan and Andrew. All right, so this goes up on a variant browser website that I designed. I'm not going to belabor this point, but these data are searchable. You can, choose how you want this data to be displayed in this table, and then it will be reflected on these histograms. You can play with that. Next steps, which I know Dan has seen, Dan and Andrew have seen. Just very, very briefly, this is something that Grace Lee and Jeffrey Schmeck-Pepper, working with Bjorn Nolman. Grace and Jeffrey went through, a tremendous amount of literature to find lots of things, including RYR2 variants and number of individuals with CPVT. And then we try to plot the penetrance density here. And so this is sort of where we're thinking of going. So this is the penetrance density that's higher and the red and thicker bands. This is a cutaway cross-section of what this calcium channel, which is in the SR, looks like. And now if you look at it from above, if you're looking from the cytosol in towards the SR, this is what that distribution looks like, and it kind of lines in these domains. And this makes some sense because this is a gain of function feature of RYR2 of these variants that are associated with this particular disorder. And so you might expect it to be somewhat more localized like this. Okay, doing good. All right, so now I've tried to put a decent amount of effort into saying, I feel like we could do, quite a bit better than just the classification. I think we can get, more precision into, at least from a basic science side. And as an aside, I don't really, it's not that I feel like we need to do away with the binary thing. I just feel like that's a step after we have as high a resolution data as possible. I feel like we're doing ourselves a disservice to discard all the richness of the data set by binarily classifying things prematurely. Although I don't think a binary classification is ultimately a bad idea. So what I've tried to point out here is that, these in silico data, in silico features, structural features are all very helpful. They help us get a prior estimate of what this disease probability is, but so are the functional data. But the problem is that our functional data are like an order of magnitude fewer than the variants that we have. So and this is the case and I would say this is a protein that's been pretty well characterized over the last several decades. So this is even an optimistic case. We're fortunate that we do have some data to go on, and that gives us enough to find a statistically significant association. But we want more. We know that it's relevant. We know that it's helpful. So we want to be able to use that. We want to have it for all variants. Okay. So it turns out that the majority of loss of function variation is due to loss of protein trafficking, and so it's most variants destabilize a protein, and that causes it to have a lower concentration. That's not all. But that gives you a nice chunk of genetic variants that you're going to find. So here, which some of you I believe are going to be familiar with, just the central dogma, you have the mRNA, this is the KB11.1 is hERG or KCNH2, that's the ion channel name. It gets produced in the ER, it gets a core glycosylation, passes through the Golgi, it has an additional glycosylation, and then it appears at the cell membrane. And you can see this process taking place by doing Western blots against KCNH2 and looking at the molecular weights. Whereas here you have the immature band at 135 and the mature band at 155. These are more, quote, benign mutations. And you see that there is this double banding, at least in HEK cells. And of note, sort of interestingly, most of them are not fully mature. So there's almost like a reservoir that's present, at least in these experimental conditions. Now, if you do a bunch of variants that are associated with the more loss of function clinical phenotype, you lose that upper banding. And then if you look at the function of this ion channel, you see a similar trend. So this loss, as you would expect, loss of trafficking causes loss of current. Okay. So now we want to design an experiment. We want to leverage that and some new technologies that I learned in the rodent lab. And Andrew was tremendous help, who showed me how to do it. And so what we did was we put an epitope tag on what ultimately presents on the extracellular face. That we can then label with a fluorescent antibody, and then we can look at confocal to make sure that this strategy is working appropriately. And so we have live cells labeled with this antibody, which gives us a nice ring at the plasma membrane. You fix and permeabilize, so the cells are dead, and you see that there's, again, as we would expect, there's some on the interior. You don't see this same patterning with variants that do not traffic very well. Again, so this is, the people involved in this is really end up being Kozak's main project. And what you can do is you can use fluorescence activated cell sorting to also get this same information. So you don't necessarily have to do confocal to get the same data. So you can see here, we have an IRES component separating KCNH2 or a wild type or variant, and an expression marker, which is mCherry. So as you get more of this mCherry, it's indicative of a lot of mRNA involved being transcribed in the cell. And then we have the live stained surface staining of that particular epitope. And so wild type looks like this, and these variants behave as you might expect. So now what we can do is we can take a pool, and what this is meant to do is that the ring around here is meant to indicate the degree of staining. So the color is, some of these like wild type are going to stain very heavily. Some of these other variants are not going to have the antibody because the channel is not present at the cell surface. We do flow sorting in bulk. We can sort all of these things and then sequence them afterwards. This is another sort of what it looks like. We separate them into these 4 bins. We sequence these bins. We come up with the trafficking efficiency that's sort of a weighted average. And so that gives us access to do this. We're limited by the number of cells that we can capture by a flow experiment, which is, in the order of hundreds of thousands. So this, again, to sort of press the point home, putting in a lot of variants are expressed in this big pool, and then it gets separated out into these 4 different bins, and then we sequence these different bins. So now we calibrate this, and we, so nonsense variants, we have very low trafficking, as you might imagine. This is in a region which I'll show in a second, just a pilot region. And then we have synonymous variants, which are actually somewhat dispersed. And this is seemingly a feature of this particular method. There is some noise here, but they're centered around 100%, which is wild type. Sorry, I should have said that before. This is normal trafficking. This is sort of gain of function trafficking. And then all the missense variants here, I think we did 11 residues or so. I'm gonna whip through this, but you get a heat map, you get variants, you get all amino acid substitutions, which I really love. I will give some reason why in a couple of slides. So you get a very rich data set. Loss of function, and I'm going to change the colors on you. I apologize in subsequent slides, but here loss of function is blue, X being the stop codon inserted here, sort of in the middle of the protein. And what I kind of want to point out here, let's see. So we did a validation. The main point I want to make with this slide is, a lot of these experiments we already kind of know. Like, you look at the in silico predictions and you can get a decent, some of these that are going to be very disruptive. And in a sense, doing that experiment doesn't really tell you anything. You already know some of this information by the in silico predictions. It's not great, but it's not nothing. Where the main feature of a method like this is the unintuitive results. That's where the experiment pays monstrous dividends. And so we did here was look at some of these that are unintuitive, like at this site. Why would a methionine not allow an alanine but tolerate a tryptophan? Why would a leucine not tolerate any kind of polar substitution except for an asparagine? And we double checked, and it looked like this high throughput experiment was faithfully producing what nature was generating. Okay, so this sort of saying the same thing. One of the features, I'd say, biophysical features. So these are the residues that were mutated, sort of broken into 2 domains here, or 2 chunks. And that patterning is based on this top half and this bottom half. And that's mainly because hydrophilic and charged residues do not appear to be tolerated very well in this top half, whereas they're much more tolerated at the bottom half. And the explanation is that the top half here is more embedded in the membrane, and so it's in a more apolar environment. So it sort of reflects some of the biophysical topology of the protein, which is great. Okay, so can we do this over a larger region? So that's the thing. The goal is to get all of them, and we are close. But this is sort of, we've done it in intermediate steps. This is work done in the lab here, and this is Rizwan's project in collaboration with Jamie Vanderbilt, which we'll mention in a second. So now we look at residues 27 to 103. This is exon 2 in KCNH2, and now we're able to get 1,450 missense variants through this experiment. Apologize for changing the colors here. Now the darker and reddish color is not trafficked, and the white or the blue is trafficking like wild type or gain of function. So again, we have the nonsense mutant right here, and you can see, and this is what the nonsense variation looks like, the distribution of all these missense variants, and then here's the synonymous variants. There are a couple down here. We measured them individually, and they did appear like wild type. So this method has noise, unfortunately. Now, comparing these data that we're getting in massive throughput, again, 1,450 different experiments, so to speak, And we're calibrating it, looking at a Western blot assay, like I showed earlier, and we see great, association. And then looking at some more recent effort, looking to characterize protein solubility in E. coli, and how that correlates with the trafficking assay is also really pretty good. Now, this is, speaks to my heart here. This is something that I, really enjoy contemplating some of the biophysical aspects of these proteins. And there are intimations of these biophysical aspects present in the heat map and the data that's generated at pretty high throughput here. One that I want to highlight here on the left is that you see this patterning, this banding patterning, which, if you're following my laser, kind of goes thick, which means more residues that are loss of trafficking, and then thin and white, which is fewer residues that are loss of trafficking. And this every other patterning is reflective of the fact that these are beta sheets that are kind of in the background here. And the beta sheet has one residue pointing up and one residue pointing down. And the ones it's too loose of a language to use, but one pointing at us and one pointing away. And the ones that are pointing at us are in this sort of collection of, or in the more dense part of the protein structure. And so mutating them away, you're more likely to disrupt the structure, which will ultimately be reflected in the trafficking. And then this one I thought was really cool. So we have a glycine 71 and a threonine 74. And what I love about this is, and you see this sort of similar, some type of patterning where it's like no substitutions are tolerated except for these two, which will be phenylalanine and tryptophan. Like a tyrosine is not allowed here. And this tyrosine 54, no substitutions are tolerated except for aromatic residues. So the aromaticity there has some influence on the structure. So there are lots of these little nuggets in here. But if you look at glycine 71 and threonine 74, it doesn't tolerate anything else. There are no other substitutions that cause the protein to traffic even reasonably to the plasma membrane. And threonine, glycine's sort of a special case. There's no side chain there, and that's somewhat unique. But the threonine, there's a serine, which also has a hydroxyl and is very similar, or valine, which is isosteric, meaning it has very similar shape, but it doesn't have that polar character of the hydroxyl group on it. And so there's something very specific about having that beta methyl group, probably constraining some of the conformational sampling of that residue. And if you look at these two residues, is right here. This is the glycine, and this is the threonine. And these are making hydrogen bonds with each other and forming this hydrogen bond network as this part of the protein bends around. It comes out this way towards sort of out of the screen, so to speak, and then makes this really tight turn and then bends back into the screen. And, apparently, and this is what's so lovely about it, is this is sort of what nature is telling us, that this is a pretty critical region in terms of the stability of this protein. Okay, so I do have a couple minutes, and I think I should be right on time. So now what we wanted, so we have a lot of trafficking results, but the gold standard is patch clamp electrophysiology. So it's it's a bit more laborious to collect. Well, I shouldn't say it that way. Actually, they both have their challenges, but we have access to I'd say about an order of magnitude greater throughput, given a similar time constraint with this trafficking method compared to even the more higher throughput patch clamp strategies. And so this was done with Andy and Jamie in Sydney, Australia, at the Victor Chang Institute. And so they characterized within that same domain all of the single nucleotide variants. So 458 variants. This is just sort of a schematic again of our strategy, generically speaking, and then their strategy, the one extra wrinkle. So they collect a lot of electrophysiology data using the syncropatch, which we also have here at Vanderbilt. But they also do this in a bicistronic expression of KCNH2. So they're looking at heterozygously expressed, where our experiment is just homozygously expressed. So there is that extra little wrinkle. And so here's a comparison of the trafficking results on the top, the peak tail current density. It turned out most of the other conduction parameters do not vary that much, or if they do, the main feature is that the current density, the peak tail current, as I mentioned earlier in the talk, is the most perturbed. And so this is, the comparison. It obviously looks pretty good. You can map the trafficking scores again. I'm not going to belabor this point because we already sort of looked at this. They have similar distributions. And I do kind of want to make the point is that there's a lot of white here. This is considered, a quote, pathogenic domain, yet over half of them appear to traffic or appear to have peak tail current like wild type. And so this is, it's a caveat to take into consideration when you're thinking about these things, what I would say in lower resolution than we are capable of at this point. Okay, so now if we look at those, so now if we take the peak tail current data as capital T truth, and we look at how well do we predict them, how well do these in silico predictors associate, and again, I'm not interested in doing something that an in silico predictor can already do. This is of little value. We want to add new information. We want to generate bits of data. And so here, looking at Revel and cardio boost, both of them, perform reasonably well in some of our other analyses with the clinical data. And so you can see we have a rank order correlation. This is looking at peak tail current density here. And so you see there is an association, it is significant. And you see for both of these, it turns out they don't add a lot of new information to each other. So they're sort of sampling the same bits of information. However, the peak tail current compared to this trafficking assay that we've developed is a significant improvement. And if you look at ROCs, which I'm not as I mentioned before, not a huge fan, but you can look at so capital T truth being sort of a random cutoff. But here we look at a bunch of different cutoffs. So these are the AUCs for these ROCs. This is a ROC that's a representative example, and here we tried a bunch of different cutoffs. And what we've done is we look at the ROC predicting peak tail current as generated by this automated patch clamp method from these in silico predictors, and they're okay. Versus just the trafficking data that we've generated alone, which is the solid black line, or when we combine them, if we use the information that's available from the in silico predictors and combine that with the functional trafficking data that we've generated, and you see there's a very modest gain. So whatever information is contained in these in silico parameters is, again, I would argue, recapitulated in this functional data set, but then there's this massive delta that we get in terms of new information learned. Okay. So, oh man, I'm like nailing this in terms of time. I thought there was no way that was going to happen. All right, so the next step here, there are several. One is we're just going to do the rest of the channel. This is preliminary. You can see because of the stop codon here generated, there's some noise. We're working diligently, or the lab is working diligently to bring that noise down. But it has, some features that you might expect that actually correlate reasonably well with what we observed previously in terms of the penetrance density. So it'll be interesting to compare these once we have the data fully available. Next step number 2 is I've had this lingering question in my mind of, do we have to do this for all proteins? Like, do we have to do these high throughput experiments on the 23,000 different proteins in order to know what their functional characteristics are? It seems to me that if we do it in this dumbest way possible, that's what we have to do. We have to do 20 or 23,000. If we do it in a smart way, we might only have to do a few thousand. But I'm wondering if there's maybe a really smart way in which we sample sparsely and can interpolate these effect maps, and maybe we only have to do several hundred. And that would be, we do have access to, we generate a lot of data. So I only, I know most intimately my lab and how much time it's taken us to do it, which is, several years, even though we're getting, we'll do all missense variants. So it's 7,000 plus, which is tremendous, but it's still several years to do. And if you do a back of the envelope calculation of a few hundred labs doing this over the, the full proteome, it's still several hundred years. So we should spend some time thinking about different strategies to maybe leverage the new things that we've learned to better predict other proteins. And so this is something that's being undertaken by an undergrad in the lab, Alex Shen, where we're looking at these other family members that are within the KCNH family, so KV11, KV10, and KV12. You can see the sequence alignment is reasonable, but not perfect. And so we're swapping out the PAS domain here and doing a full mutagenesis experiment there to really try and figure out how much, if we do KCNH2, can we predict how much of this high throughput trafficking experiment can we predict for KCNH1 to try and get a sense for how divergent can the sequences get before we lose that new information we learned. All right, the last thing that I wanted to get to is, I've spent a lot of time working on and thinking about these single variants, variant properties, and how we understand them. But these variants occur, quote, in the wild. There's a context there. It's not, they don't occur in isolation. And so the question is, and Dan can speak to this because I know he's thought about it a lot, is, all right, well, you do it, you could make that edit, you could put it into an IPS line, an induced pluripotent stem cell line, make cardiomyocytes and investigate the electrical properties. And there's, the field is getting better at doing that for this particular disorder. But then the question is, well, okay, have we done it enough? Like, we did it in one person, but do we have to do 10? Do we have to do 20? Do we have to do 100,000? And so, who is like, quote, representative? And so this is, what my lab is really pivoting hard into. Devin is the one who's really enabled us initially. Now everybody's doing it. And we're getting cells from Ben Shoemaker, and we're working with Quinn to try and get access to BioView, which I think is the most reasonable resource for this particular approach. And I'm just going to describe it here in a couple of slides, or really just one. So the idea that we've had is that most of these variants that are different between individuals are going to be meaningless in terms of the phenotype we're interested in. However, some of them may have an influence. And I think a reasonable place to start is to look at what we've already seen for several years now, which are these GWAS results. And it's a topic that is being fervently debated right now, is what to do with these polygenic risk scores, and many of them appear to have effects that are of the order of these single Mendelian high effect variants. And so what we're trying to do is collect people. So this is higher polygenic liability lies to the right. And we're trying to take individuals, and this is for us, I mean, it kind of doesn't matter, but for us, for right now, is QT interval. We're taking people that are on the extreme low end, the extreme high end, and then a representative in the middle, and making the assumption that the variability that exists, the relevant variability that is going to be out there, might be covered to some extent by this variation. And so what we want to do is we want to take these variants that we are understanding at a very deep level in isolation and put them into these different groups and see what is the variability of the phenotype. How is that phenotype varied under the influence of these distinct genetic contexts? And this is so we can do a lot of things. We can do, transcriptomics, protein interactome stuff, and patch clamp and the EFPs. But then we're also looking at this in some of the clinical populations through the collaborations that I mentioned at the onset. But the hypothesis being that, it's going to be mixed. You can't take them all together. I would argue that the variants that we think have a huge effect size, the polygenic background isn't going to matter as much. Whereas the variants where the effect size is more modest we would expect, a greater input or greater variation due to the polygenic background or genetic background in general, but we're thinking to start, it would be the polygenic background. And so we started doing this. This is, somebody at the 99.18 percentile, an individual at the 0.35 percentile of the QT PRS, and someone right in the middle. And so we're in this, and this is sort of ongoing. I believe I'm like, oh, I am a touch over, but this is where I wanted to get to. So those are the directions that we're going. That's sort of where we've been. I've tried to highlight some of these people along the way, but it's obviously a collaborative effort. It's one of the things I enjoy most working here at Vanderbilt is the amount of collaboration and the access to these different groups, and then the funding in the lower right. And I will take questions.
Your samples are all from Japan or Europe. Any idea about how to extend your sampling to other parts of the world, South America, Africa? You thought about that?
So we did try to compare how our the prediction performance, both with our method, but all other in silico methods, and how that might vary between variants that we see in both the Japanese population and the Europe population. And we weren't powered enough to see any differences. I mean, there were, but they weren't statistically significant. I've tried to take the position of ideally what I want is I want all variants in these genes that we know something about that have ever been seen, that have ever had any clinical phenotype. I'm really being agnostic to their origin to first order. I think sort of starting at that point and then trying to layer on, how does the background work, as sort of what I alluded to at the end in terms of PRS, but then obviously the ethnic differences as well. So I guess I've sort of punted on that, you could say. I think, I'm just trying to get as much data as possible, and then parse it out as we are powered to do.
Well, we've had this long debate in our lab about what is an African origin IPS look like, and how is that different from a European origin? And you can argue yourself into amazing circles and then get nothing done.
Yeah, exactly. And this is I drove down to Jackson, Mississippi to try and talk with the Jackson Heart Study people. Something that Quinn and I have been talking about is trying to do DCM. And I think it obviously would be a huge undertaking, but I think if you could do something like what I mentioned in the previous slides about the PRS distribution and look at rare variant, or even just the regular or the native interactions, and when you take the PRS, and I mentioned DCM because we know it's significant in the African ancestry, which isn't always the case. If we did that and then compared it to the European ancestry to sort of try and just to get a starting point, to see what are the things that might be more different or might trend to be more different.
Other questions? I think we're running late, so if you have questions for Brett, shoot them by email, I guess. I need to go to my next meeting. Brett, that was terrific. A real breathtaking journey.
Thanks, Dan.